All right. So, good evening, everyone. My name is Parth, and I thank you all for joining with us today. Let me introduce to our speaker, Dr. Yatish Turakhia. Dr. Yatish Turakhia is a postdoc researcher at Genomics Institute, UC Santa Cruz, where he is working with Professor David Hustler and Professor Benedict Payton. He is going to join University of California, San Diego next year as an assistant professor in the Department of Electrical and Computer Engineering. His research interests include developing hardware accelerators and algorithms for faster and cheaper genomic analysis. Dr. Turakhia obtained his PhD from Stanford University in 2019, where he was jointly advised by Professor Bill Daly and Professor Gil Jarno. His work has won Best Paper awards at ASPLOS 2018 and IEEE Microtopics Award 2018. He is also a past recipient of NVIDIA's Graduate Fellowship. So in the first of the talk, Dr. Turakhia will be presenting the designs of his earlier coprocessors as a case study and show that a domain-specific hardware can provide massive speedups for genomic application. In the second half, he will be showing the usefulness of this approach to solve some emerging problems in biology and medical science that includes fight against such pandemics, the current one and upcoming in future, probably. So at the end, we'll hold a Q&A session for half an hour or 20 minutes. So please pile up your questions beforehand. Now I request all the attendees to please mute their microphones and we don't have audio disturbances. So without boring you further, I hand over to Dr. Turakhia.
Thanks a lot for such a wonderful introduction. Hi, everyone. Thanks a lot for joining. As you can see, I'm actually be joining UC San Diego next year as an assistant professor. So just a little bit of heads up. This talk is going to be partly technical and partly also a sales pitch with the idea of giving you the broadest possible overview of what I am doing in order to convince you to hopefully apply for joining the lab maybe next year or even in future. Um, so with that said, I'll begin. One thing which I would really like to share is like the trend of the growth of genomics data in recent years. Now, genomics data has been witnessing an unparalleled data explosion and has been growing at about 125% per year in recent years. This means that it has far outpaced Moore's law and is on a trajectory to grow faster than any other data type in the world. In fact, in sharp contrast, the CPU performance scaling for which the Moore's law was originally coined has slowed down considerably in recent years, as you can see from this plot, and is now taking place at just 3% per year. Now, the technology that has spurred the massive explosion of genomic data is known as DNA sequencing or genome sequencing. The idea is as follows: genome is the basic blueprint of life and is the sum total of all the information that's contained in the DNA molecules in all our cells. And what DNA sequencing does is it's the technology that enables reading of raw nucleotide bases from a DNA molecule. So what I'm showing here is a DNA sample which is that sample, by the way, could be from like saliva or blood, which is fed into a machine which is known as DNA sequencer, which produces a set of output reads. These are called sequencing reads. And these are just sequences of nucleotide bases from a contiguous fragment of DNA. For example, this fragment could be from the region which is underlined above as shown in this figure. Now, the thing is, the modern DNA sequences come in a variety of sizes and forms. So for example, you can have like these rack size sequences from Illumina and Pacific Biosciences that can sequence several tens of human genomes per day. But you also have like these Oxford Nanopore Minion type handheld devices that take several days to sequence a single human genome, but are portable. And this has created a revolution in actually genomics because as you can see, for example, in 2017, this device was actually taken to space by NASA to carry out the first DNA sequencing experiment in space. This, by the way, is Kate Rubins, who was recently also in the news for being the first person to have voted from space from the ISS. We've also been doing a lot of extremophile sequencing in Antarctica to study what type of extreme organisms are able to survive in harsh environments. And more recently, during this pandemic, and I'm going to talk about this later in much more detail, but through the use of such devices, we've been able to actually generate 100,000 SARS-CoV-2 sequences, and these are full sequences. So this is not like RT-PCR or something. This is like exactly what RNA code does this virus carry. And we've been able to do this at a scale which is completely unprecedented. Many people find that the growth in genomics data is quite astonishing, but they also believe that this growth will soon come to an end. Since there are just a finite number of humans on Earth, there's only a finite amount of sequencing that's to be done. This is actually not true. Not only are we a long way from any point of saturation, but with the new application of genome sequencing that are emerging, there will never be enough sequencing. So consider this, that there are over 7 billion individuals on Earth, of which not even 0.1% of them have been sequenced. Sequencing individuals can inform us about their ancestry, their physical traits, disease propensity, and much more. And so the more the number of individuals that we sequence, the more we are going to learn about each individual. Oh, sorry. So this is why the governments of the United States, European Union, as well as individual provinces of China have all announced plans in recent years to sequence over 1 million individuals each in the coming few years. And if that isn't enough, there are over 100 million species on Earth besides humans. Sequencing more species helps us learn how different species are related to each other and what in the genome of one species makes it different from another, and how genome influences specific traits such as flight or development of eyes and so on. And this is why large-scale sequencing efforts are now planning to sequence millions of species over the next decade. And if that isn't enough, personalized genomics is now also entering agriculture and pet industries and is transforming genetic engineering. Many startups and companies are now working on perfecting a technology which is known as liquid biopsy. And this involves sequencing of circulating DNA fragments in our bloodstream. Essentially, this can be used to diagnose several infectious diseases, such as sepsis and COVID-19, down to the specific strain of the virus or pathogen that's causing the disease. Previously, we used to use blood cultures to do something like this and would take several days. And those are going to become a thing of the past because sequencing is much faster. Liquid biopsies can also be used to non-invasively diagnose somatic mutations. So these are mutations which occur in our cells during our lifetime and can potentially cause cancer. So for example, a mutation uh, causes a cell to divide indiscriminately, and that causes the tumor of the cancer. And this can be done with the help of liquid biopsy. The diagnosis can be done long before the cancer actually gets dangerous. And so in future, I think many of us are going to get sequenced not once, but on a routine basis. We've also now begun to understand that our bodies are full of microbes. There are perhaps more microbial cells in our bodies than human cells, and they play an important role in everything from our digestive system to our immune system and even our nervous system. Understanding the genetic makeup of our microbiome will inform future healthcare and drug therapies. Besides that, many researchers are now using sequencing to understand how the same copy of a genome present in all our cells has instructions in it to develop into hundreds of different cell types in our bodies, such as muscles, brain, liver, and blood. Now, again, this application requires tremendous amount of sequencing across space, which means that we'll be sequencing a lot of different cell types and time as well, which means across different stages of development. And if all of this wasn't enough, today genome sequencing is also used to greatly influence our species conservation efforts. For example, it is now possible to bring home a sample of water from a nearby lake or river and sequence the floating DNA in it. And that can be used to monitor the ecosystem biodiversity as well as migration patterns of species in a completely non-invasive manner. There are perhaps a dozen of other examples that I might have skipped, but what I'm trying to do here is try and convince you that as sequencing continues to get cheaper and more accessible, more such applications will emerge. It is also important to realize that as Moore's law is coming to an end, the computational costs for such type of applications would pose great barriers over time, lot more than the cost of sequencing itself. And so I'm really motivated in my research to enable the vast potential of genomic data. And so I've sort of made it my mission to sort of eliminate the computational barrier that exists and would exist in genomics. So broadly, my research interests fall at the intersection of algorithms, biology, and computer architecture, as this Venn diagram shows. But more specifically, I'm interested in developing algorithms and domain-specific hardware accelerators that enable cheaper and faster progress in biology and medicine. So to that end, I will be covering 6 of my previous research projects in this talk. The first 3 are actually based on hardware accelerators, that's Darwin, Seg-Align, and Darwin WGA. And last 3 are actually applications of those type of alignments, for example, to make biological discoveries. And that includes Usher, which we are using for COVID-19 phylogenetics. We've done a convergent screen, which was published in PNAS last year, as well as finding gene losses in mammals, which we published in Nucleic Acid Research earlier this year. But before I begin, I would first like to acknowledge my PhD advisors. As you can see here, Professor Bill Daly and Professor Gil Bajirano, as well as my postdoctoral advisors, David Hausler and Benedict Payton. And I've been fortunate enough to be able to collaborate with a bunch of people from various organizations that includes Microsoft, PacBio, NVIDIA, Stanford, and UC Santa Cruz. And the co-authors on my papers have been actually highlighted in bold over here. In fact, some of these also share first authorship with me on the papers, which I mentioned before. And I would also like to thank a lot of these funding resources who have supported my research. But specifically for this event, I would also like to really thank Vedant, Yagya, and Mohana, who helped in arranging this talk, as well as Professor Sachin Patkar, who had originally invited me and who was also my DDP advisor at IIT Bombay many years back. So with that said, I'll begin with Darwin. And so this is going to deal with an application of long-read assembly and had appeared in ASPLOS in 2018 and also won the Best Paper Award there. So I already told you about DNA sequencing. However, what I've not told you yet is that DNA sequences are also error prone. And so the resulting read sequences that you see here are actually a bit different from the original DNA fragment from which they were sequenced. So what I mean by that is they could have some number of bases that are deleted, some which are inserted, so they never existed in the original sequence, but just seem to appear, and some which are substituted, which means that a C was wrongly interpreted as G, for example, over here, and so on. But the good thing is the errors tend to be mostly stochastic in nature. And so they can be fixed or lowered by producing a consensus of multiple aligning reads. So for example, in this figure, I'm showing 3 reads which originate from the genome sequence as shown above. And the reds are showing the errors that occur in those reads. And as you can see, if I take the consensus of these aligning reads, then the consensus has a much lower error rate than the original reads. So this process of finding alignments of multiple sequencing reads to produce a final consensus sequence is what, in a very simplified manner, is known as read assembly. So long read sequencing is basically a new technology in DNA sequencing, which enables us to read long stretches of DNA at once. So previously, we used to be able to do only short fragments of DNA, only 100 base pairs or so. But now we can do 100 times as large, except the problem is that the error rates are also higher for this technology. Now, I should mention that this has improved in recent years, but the results which I'm going to show hold true at the time that Darwin was published. And it is because of this high error rate that the cost of assembling the reads using long reads is also very high. So for example, it could take up to 5,000 CPU hours when a reference is used to guide the assembly, and up to 60,000 CPU hours when we do it de novo without the benefit and bias of a reference sequence. And by reference sequence, I means that we just align it to a single individual. That could be someone like Bill Gates, for example. And because his genome is likely to be very similar to ours because we are all humans, uh, we could actually align and just see the differences with respect to him uh, to identify what mutations, for example, some of us carry. And de novo doesn't do that. So it's actually much more expensive to do it uh, that way. And as you can see here, it's actually an order of magnitude higher in computational cost compared to short read assembly. So why do we even do this? Why do we even use long reads? And that's because despite their high error rates, they are considered to be the holy grail of sequencing for a very long time. And that's because they have an ability to discover a very broad spectrum of genomic mutations, such as structural variations as follows. So they could be something like a tandem duplication, inversion, and so on. I won't get into details, but I'll just point out that some of these mutations we are now starting to believe uh, are also likely to cause like some very debilitating diseases, including cancer and autism. They are also very superior in doing a haplotype phasing. So what I mean by this is they help us in identifying maternal and paternal mutations. So you all know that we actually carry two pairs, like a pair of chromosomes, one from mother and one from father. And with the help of long reads, we are actually able to phase it, which means we can identify that A and G, these mutations actually occurred on the mother chromosome, and T and C over here occurred on the father chromosome. That's not really possible with short read, where usually the variants are unphased. They also help us in resolving repeats, which are quite abundant in the human genome. So basically, the human genome, almost 50% of it is like repetitive sequences, and they are really hard to be resolved using short read sequencing. So for all of these reasons, we actually decided to develop a coprocessor, which we call Darwin, um, to essentially speed up the process of long read assembly. And essentially, it consisted of two novel algorithms, DSoft and GACT, both of which were hardware accelerated uh, and interacted with the software using a very flexible API. So it's programmable in a way to handle different types of long reads. I won't be actually going into the detail of these algorithms, but what I'll tell you is DSoft was essentially like a filtration algorithm. So essentially, what it did was to actually reduce the search space by up to 1,000,000 fold for a more computationally intensive GACT step that essentially was a heuristic to Smith-Waterman. Uh, and its key feature is that GACT was a novel algorithm in the sense that it used constant space to do compute intensive step of the alignment. And I'll show later that this has profound implications for hardware acceleration, and that would be highlighted later. But in the interest of time, like I said, I'm going to skip the details of the algorithms, but actually point out to you like what are the main lessons that we learned and what were the real sources of hardware acceleration that gave us large speed up using Darwin. And this is basically the sort of tricks that, if you were to design a domain specific hardware accelerator, you would probably be using some of the tricks from this slide. And then I'll give you some concrete examples how we used it in the context of Darwin. So number one, we did hardware software co-design. The idea is simple that you do more of what is easy to accelerate and trade that off for less of what's hard to accelerate in hardware. We also use specialized operations, which means that we did in one cycle what in the custom hardware, what would otherwise take tens of cycles on a conventional processor. We exploited parallelism. Now this might sound obvious, but actually there are lots of levels of parallelism that you can exploit. There could be inner loop parallelism, outer loop parallelism. And it's important to both exploit as well as balance parallelism at each level. Uh, and we also use local memories. And as many of you guys would already be aware, the small and local memories like caches on the SRAM tend to be order of magnitude better in both energy and bandwidth as compared to DRAM. We optimize global memory accesses, which means that DRAM, because it favors more sequential access pattern. So we optimize our data structures in a way that, we did more of that. And finally, we reduced overhead, which means that because we are using a specialized processor, we don't have the overhead that's otherwise incurred by a general purpose processor on program interpretation. So I'll now explain very briefly, actually, how these gains came up in Darwin as an evolution of steps from graph map. This is the state of the art, used to be the state of the art aligner for long reads. And I'll show that as an evolution of steps where the time is broken up into these filtration and alignment steps. And you can see that the time is shown in logarithmic scale over here. And I will evolve graph map into Darwin through those steps and show you the speed up at each stage. Uh, so GraphMap, like most other aligners that we have today, spends most of its time in this filtration step. As you can also see, that's in blue over here. Most of the time is spent over here. And the reason it does that is because it's actually trying to avoid doing more of alignment, which is actually more computationally intensive on the processor. Now, this tends to be the right thing to do in software for high performance, but is not very good for hardware acceleration because the filtration step is memory bandwidth limited, and you use the same memory no matter whether it's the CPU or a specialized processor. So you face the same constraints. So the first thing that we did in Darwin was to actually replace the filtrations and alignment steps by what I called earlier DSoft and GACT algorithms. And what DSoft does is it actually spends 400-fold less time on the filtration steps, but in return, it has to do 500 times more number of alignments using GACT. And as a result, it actually results in a counterintuitive slowdown of about twofold when it's implemented in software. But the good news is, remember this time is on a logarithmic scale, that an overwhelming amount of time is now being spent on the alignment step. And that's going to be really easy to accelerate in hardware. So the first step that we did for acceleration in GACT is to implement specialized operations, which means that GACT was actually required to compute like a matrix, uh, uh, a series of large number of matrices like this, using the help of dynamic programming equations, which are known as Smith-Waterman-Goto equations, as I'm showing here. But when you implement these equations on a modern CPU, what you typically tend to find is that computing these equations for a single cell would require about 37 cycles and 81 nanojoules of energy. Now, if you implement it on an ASIC, let's say a 40 nanometer on a 40 nanometer technology, what you'll find is that you can actually implement all these specialized operations and do it in a single cycle, uh, because they are just a bunch of adds and comparators put together. So that itself results in 37 times speed up. But the other good news is that you could actually spend much less energy doing that because you only spend about 3.1 picojoules of energy. So that's about 26,000 fold gain. And you might wonder, where is this gain really coming from? Of course, some of it is coming from specialized circuit, which uses low precision arithmetic to do these operations. But a lot of the gains is actually coming from the fact that we are reducing the overhead. That was point number 6 in my list. And so if you look at a typical CPU, if it does one add instruction, uh, I guess many of you will find this surprising, that over 99% of the energy in the add actually goes into program interpretation. So that's all the way from instruction cache access to register file access to control operations and so on. And a very small amount of energy is actually spent in doing the actual add. But when you implement this on an ASIC, you almost completely eliminate that overhead of program interpretation. And as a result, you get significant improvement in energy efficiency. Well, this figure is, I've taken that from Google's TPU paper, but it's also representative of Darwin. But once there are huge energy savings, the obvious next thing to do is to add parallelism. And we added like parallelism in the form of wavefront. I'm not going to go into details, but it essentially exploits some parallelism, which is fine grained using these elements. Uh, and because we supported traceback, we actually used SRAM memory to hold the traceback pointers actually. And using SRAM as opposed to DRAM saves you 2 orders of magnitude in energy efficiency. And so that's huge. But the reason why we were able to do this, use SRAM instead of DRAM, which is what other algorithms would use, is because we did some co-design. And this is a classic example where co-design could actually enable you to use local memory. Uh, so what we did was we actually modified the alignment algorithm into a heuristic in a way that we computed this really long alignment as a series of overlapping tiles, where for each tile, you're just storing the traceback pointers for that particular tile, which means it's actually constant memory as opposed to previous algorithms, which are all linear, at least linear, in the amount of their memory requirement, which means for long reads, they had to access DRAM memory in some cases at least. And so once you've reached the limits of inner loop parallelism, you can actually exploit outer loop parallelism. So in DNA assembly, for example, you have millions of these independent reads that could be aligned together. And because we actually saved a lot in energy efficiency as well as area by using these processing elements instead of general purpose instructions, we could provision a lot of them on a chip. So in Darwin, for example, we provisioned 64 of these GACT arrays. Uh, and they all exploited outer loop parallelism in the form of independent reads. Now you just have to balance inner and outer loop parallelism for maximum gains, because if you keep exploiting outer loop parallelism, you will actually hit uh, the amount of SRAM memory budget that you have, whereas it does not grow with inner loop parallelism. Um, so that's important, and that's what we did in Darwin. So all of this combined, we actually get 80,000 speed up on GACT alone, which means that we got 380x speed up on the assembly. And now the bottleneck was shifted back to the filtration step, DSoft. So the next step, what we had was to actually accelerate a DSoft. And now, again, I'm not going to go into details, but essentially the point of our accelerator was to, what we did was that some of these data structures which had random accesses, we actually shifted it to SRAM, which means that you could access those random uh, memory in in constant cycle, as long as in a single cycle, as long as they're on separate DRAM SRAM banks. And so that gives a huge speed up. But because they are random, you actually have to route the updates to those memories using a network on chip. And the large data structures, which were mostly sequential in sequential access pattern, they were still on DRAM. So we not only reduce the amount of DRAM accesses that we do, but we also improve it to sequential access pattern. And that in turn gave us another fourfold speed up, just because we use more amount of DRAM, and another 15-fold speed up because we reduced the amount of memory accesses by threefold, and made it sequential in nature. Uh, and finally, we did a very common technique that's used in hardware acceleration, which is to do pipelining. So DSoft and GACT could actually be pipelined for reads, and that gives another 1.4 fold speed up. And if you combine all of these together, that's about 15,000 fold total speed up. Uh, and so we actually analyzed in ASIC using design compiler on a 40 nanometer TSMC technology. And the area and power numbers are shown over here. And as you can see, the area is dominated by, as well as power is dominated by SRAM on this particular chip, and not the logic. For baseline, we had used like a state-of-the-art Intel Xeon processor, but a single thread, because that's like the ISO power point of comparison, especially after you account for technology node difference. And these are the performance numbers for different long reads. But as you can see here, the speed up is actually huge, 1,000 to 15,000 fold for reference guided assembly, and about 700 fold for de novo assembly. So the reception that we got for this paper was really, like, we were thrilled to see that. We were not only conferred the ASPLOS best paper award that year, but we were also selected for micro top picks, which is very prestigious and highly selective as well. But besides that, we had like extensive media coverage. For example, we were covered by the Next platform as well as the morning paper. And I was invited to give a number of industry talks that includes at NVIDIA, Qualcomm, Microsoft Research, and Pacific Biosciences. This is, by the way, a sequencing company. And in order to have real world impact, I actually worked over a summer with Microsoft Research. And after that internship in October 2 years back, they actually made this announcement prior to the American Society of Human Genetics meet in 2018, which is where we also presented this work. And you can read this announcement. They say that this technology was really like a completely new method. And by the way, this was implemented on their Catapult system. So we utilized FPGAs for this. And as you can see, they are also calling it revolutionary genomic service that they hope to provide widely to the community. Besides that, like, my advisor was invited to give a keynote talk at one of the HPCA workshops. I was invited to give talks also at USNIC's ATC and International SOC conference. And we were also featured on the CACM cover page that year. As you can see, this is our article. So this article followed Darwin, and we used major insights, especially the 6 points which I highlighted in my slide earlier. They are mentioned in a lot more detail over here. As you can see here, I'm the second author on this paper on this magazine. So that covers Darwin. Now I'll actually move on to Segaline, which is actually going to appear in supercomputing this year, next month. But it covers the application of whole genome alignments. So before I explain Segaline, I'll actually have to tell you what comparative genomics really is. This is a field where, as the name suggests, it involves comparing genomes together. And in this case, it's actually comparing genomes of different species. And that has a lot of applications, which I'm going to highlight later. But one thing that's important to note is that the appetite of sequencing in this space is never ending. It's ever increasing, actually. And this is the reason why there are many consortia right now ongoing, such as the Earth Biogenome Project or the Vertebrate Genome Project. Each of these are like multi-million-dollar government-funded projects, and their effort is to sequence tens of thousands of new species over the next few years. But because on their own, these genome assemblies, like just the sequencing, won't inform you much. It's actually when you compare uh, the genomes of different species, can you actually make fascinating inferences. And so that's where the comparative genomic applications comes in. So for example, you can compare genomes of different species to do phylogenetic inference, which means try and understand how different species are related to each other. For example, now we really understand very well that chimp and bonobo are the closest relatives to human using just the genetic code. And they are actually more closely related to each other than they are to human. And the next closely related living ape is gorilla, and then followed by orangutan, and so on. But this something like this could be done for any species that you can think of. And this is a very important application to try and understand evolution from a molecular perspective. Um, you could also do a lot of other interesting things. For example, it could, as you know that our genome, I probably know that our genome consists of like genes, and these genes code for proteins, and they are really functional. They are the workhorses of the cells. Uh, so what you can do is once you look at the genomes of different species, and we've sort of evolved from a common ancestor. That's the Darwin's um, uh, uh, theory of of evolution and natural selection. So what you can do is you can find the conserved regions of the genome, and you can use those conserved regions to help annotate what are the functional elements. For example, in this case, it's actually the protein coding gene, and all of this is actually achieved uh, through this comparison, which is done with the help of whole genome alignments. And so that's something that the entire field of comparative genomics is actually relying on. Now, the interesting thing is, in recent years, I think the whole genome alignment application in comparative genomics has really entered the supercomputing scale. So just 20 years back, in 2001, we had sequenced through the Human Genome Project, the very first human. That was in 2001. And by 2009, we had about tens of species. For example, we had mouse, and we had vertebrate species. So we had rat, we had gorilla, and so on. Uh, and that has been increasing over the last decade. So just this year, if you see, we've actually crossed 1,000 vertebrate species already. Note that this is an exponential scale, and it's projected to grow really fast in the coming years as well. Uh, so why am I saying that it's entered the supercomputing scale? Because just last year, actually at UC Santa Cruz, just before I joined them, they had actually done a 600 realignment. Those were the number of species available that year. And they had compared those 600 species together using whole genome alignments. And it actually took them 81 days on a massive cluster consisting of 80 CPU, 800 CPUs. Now just project that to 2025. And what you'll realize is that the same thing would actually take more than 3 years for the number of species that would be available in 2025, which means that the assembly is not going to be the whole genome alignment won't even keep up with the assembly rate, because more number of species are going to be sequenced that year. Then actually you can align together using whole genome alignments, at least on this cluster. So you need really supercomputers to be able to do that. um, and so in order to help, we actually decided to accelerate this application. And again, I'm not going to go too much into details. This is the system overview. You can actually read that in the paper. I've already had it on my website. But the essential idea is that we had to optimize both CPU and GPU implementations. This was a GPU accelerator. So one particular stage had to be accelerated on the GPU, that seed and filter, and the rest of these are actually parallelized on a CPU. And we also have a multi-node implementation, which means that you're not just processing on a single server, but you have like, and this, by the way, was done using Apache Spark on Amazon Web Services. And you can easily parallelize this particular application and get results really fast. But the bottom line is that our implementation resulted in 13 to 14 fold speed up over state of the art LASD tool, as you can see here. And we have achieved actually pretty good scaling efficiency as well. So even if you increase the nodes to like 64, we actually still see more than 80% scaling efficiency. Uh, and by the way, this just to show you that this has had impact, we actually are currently working with NVIDIA, and they have this GenomeWorks library, which is for genomic applications, as you can see here. And this is the team that we are collaborating with. And they've now integrated Segaline's XDrop kernel into this library. And it's currently in the development mode, but it's going to be in the release version very soon. Uh, and the paper which I mentioned to you before, the 600 realignment, which was carried out last year, they have a bioarchive preprint, but this year it's actually appearing in Nature. So it's the best journal you can like ranked usually the #1 journal in the world. So it's a very, very high impact journal. So this is going to appear probably next month, I think. I'm not on this, by the way, but we are now working on with the same team at UC Santa Cruz, doing 1,000 species this year. And the good news is we are actually going to be using Segaline to do this because it's just too prohibitive to do it anywhere else right now. Uh, so that's Segaline. Now I'm going to move on to Darwin WG. Now this is actually the same application. It's the same whole genome alignments, and we're going to compare to LASI. So I don't need to give you the background for it. We actually published this in HPCA last year, but this is different from Segaline in the way that what we had noticed with LASI in particular is that it had some sensitivity issues, which means it was not able to discover some of these alignments, which we thought are very useful. And people have published about this extensively in the past. But the basic problem is that LASI-like tool or Segaline now would work really well when you're comparing closely related species. For example, if you're comparing human and chimp together, they are already very similar sequences, so it works very well for that. But as soon as you move very far away in evolutionary time, for example, human and mouse, which have deviated 88 million years back as compared to 6.5 for human and chimp, the differences between these genomes tend to be high. And so you miss out on a lot of interesting alignments, which could result in biological discoveries. And the way to increase the sensitivity we've already known is to replace one of those gapless filtering stages in that algorithm by gap filtering, except that no one does it because it actually slows down already slow software by 200-fold. So it's really prohibitive to do it. But what we showed in Darwin WGA is that you can actually do it pretty fast on an FPGA, and this is some of the insights which we had already derived from Darwin. And so what we did was we actually replaced that gapless filtering stage with gap filtering, which increases sensitivity, but was also fast. So by the way, both of these stages were hardware accelerated because you had a lot more alignments to do. So even that needs to be accelerated. But yeah, we show in the results that we are a lot more sensitive compared to the state-of-the-art tool. And that's useful, especially when you are trying to compare distantly related species. And we showed like an FPGA prototype that was working on AWS F1, and we like with all our tools, we actually released it on GitHub, including the RTL for this design. And we implemented this on this particular FPGA. But we also did the same way that we did for Darwin, an ASIC design compiler analysis. And as you can see, compared to ISO-sensitive software, we are much faster, although we are slower on the FPGA for compared to LASI, but that's also less sensitive. And we match or exceed by a little bit with ASIC. So you can do basically in the same time with an ASIC that you do on a CPU, but get more sensitive alignments. So that's the point of Darwin WGA. Now with that, I'm actually going to cover Usher, which is one of my most recent works. We've released the bioarchive preprint just this month, earlier this month, and it's currently under review in Nature Genetics. But it deals with COVID-19 phylogenetics, and I think some of you guys might actually find it interesting. So as you know, this virus is actually quite nasty, and one thing in pandemic that we often want to do is to track how this virus is evolving. So this virus is dividing, and as it's dividing, it's actually also acquiring some mutations. For example, it didn't acquire any mutation. It's the same virus when it replicated once, but in this particular replication, it actually must have acquired a mutation as like a different strain of virus, for example. in this case. And that could be happening with COVID-19. And you could have very dangerous strains as well, something which is more transmissible or something which is more lethal. And you really want to track it. And what's happened with, the revolution of genomic sequencing is that it's for the first time, that we've also had like an exponential growth, and we've been able to track this particular virus at a whole genome level, uh, um, at an unprecedented global scale. So we are continuously monitoring right from the first virus, which was actually released publicly by the Wuhan lab on 24th of December. And earlier this month, actually, we actually crossed 100,000 sequences. This is totally unprecedented. It's never happened. Even a fraction of this was not done for Ebola or previous SARS and so on. But what this is enabling is what we are calling genomic contact tracing, which means that now you are able to trace the local transmission dynamics of this virus. You can also infer with the help of these sequences how many new introductions happened for that virus in a particular country or area. So, for example, you can infer that about 40 times in India the virus actually was introduced from Iran. About 15 times it was introduced in UK or from Australia, and so on. So you can get idea of those. Where is the virus coming from, and how much of it is just about internal transmission? How much is coming from outside? And so on. But you can also use it for transmission chains. So let's say a bunch of people got infected in a particular locality. You can actually try and infer using phylogenetics who could have been the first person to have been infected, and how was the transmission chain among those infections. And you can also do it for unrelated infections. So, for example, one person in US and one person in India both deposited their sequences in this database, and you see that they are identical. That means they must have been in contact at some point, and that's otherwise not really possible easily with other contact tracing methods. But with genome sequencing, it's actually pretty easy to do. But the question is, can we actually do this all in real time? That's actually the key goal uh, in contact tracing, right? You want to do it really, really fast, and that's where hardware acceleration could play a very important role. So, for example, currently, if you want to, like, for example, let's say some new sequence was available from India, and they want to, analyze it with all the global sequences that are otherwise available. What they would do is, they would add that sequence to the whole database. And this is the current phylogenetic tree, how the virus has been evolving in those existing sequences. And then they would use something like a placement tool to sort of identify where is this virus, like where should I place this particular virus in the context of this global phylogeny. And that's really the key task that people want to do, like virologists, epidemiologists, and so on. Uh, but unfortunately, with the currently available tools, this is actually really slow. It takes tens of minutes uh, to do it for a single sample, by the way, just one sequence. And it also takes a lot of memory to do this with the currently available tools. By the way, I'm talking about only 38,000 base tree. Now we have, like I showed you, more than 140,000 sequences. So it's going to be a lot slower. But this is nowhere close to real-time constraints, right? Um, so what we did in our work at UC Santa Cruz is developed this tool called Usher, which stands for ultra fast sample placement on existing trees. And as you can see, Usher is way faster. Like it can do it in half a second, what takes several tens of minutes. And by the way, this is a completely software implementation. There's no hardware acceleration involved here, but you might ask, how is this even related? I actually used a lot of computer architecture concepts to actually accelerate this. For example, how to use cache efficiently and so on. But as you can see, this is really, really fast. But more than that, it actually scales pretty well as you use more number of threads. And so, although you can even use this on a laptop, but as you have more powerful servers, you could, this, by the way, I'm showing for 1000 samples. And you can place 1000 samples in only 18 seconds with 16 threads. So it's way, way faster. And what that has enabled us to do is, by the way, we've developed this tool, and you can actually go to this website and check it out. But essentially, there's a genome browser that we've developed for SARS-CoV-2 as well, which means that you can do everything on a web interface. So we provide we do it on our own servers, all the analysis. People can just upload their sequences and get the results back. But essentially, what this browser is showing you is this phylogenetic trees for the SARS-CoV-2 with genomic coordinates shown over here, the mutations that the virus has shown in red, and green over here, red which are more functional in nature, and green which are more like benign mutations. And the newly placed samples which you might have uploaded are highlighted in this blue over here. And you can do rapid cross-referencing with like several biology tracks on this particular browser. And I'm happy to tell you that now there are a lot of people who are already using it, including some people from Los Alamos National Lab. Uh, with that said, I'm actually going to speed up a little bit now because I also want to move on to like what's the future work. But maybe I'll just quickly mention that besides developing co-processors, we've also been involved in doing a lot of biological discoveries as well. Especially because at least at Stanford, I had a co-advisor who was really interested in doing that. So one application that we did biological discoveries for was actually convergent evolution. Some of you guys might have heard about this, but this is a phenomenon which is really fascinating in biology, where a common trait is independently acquired by more than one species. It's like a very fascinating thing that happens in nature. For example, one of them is echolocation or biosonar. And this is one of the most striking examples uh, uh, of convergent evolution and has been a subject of many books and papers in the past. So whether it's hunting inside murky waters for dolphins or in dark skies for bats, both of them actually use the same technique, which is that of echolocation, right? So people have wondered, what is it? This must be genetic, right? Like, what is it in their genomes that must have led them to acquire this common trait? And people have also wondered whether it has resulted out of like common or identical mutations that these have acquired in their lineages. This is, by the way, the evolutionary tree, so you can see that they are independent. So what I mean by convergent mutations is, for example, there was a amino acid L over here in one of their positions, and they both independently changed it to a letter M, and that helped improve their hearing or something independently, and really helped them develop like a trait like echolocation. So, in other words, does molecular convergence underlie phenotypic convergence? And this is one example of convergent mutation. But another could be like a strict convergent mutation. This was parallel, where in this case, different amino acids actually converge to a common amino acid, and that's also possible. And that's opposite of divergent mutations, where you started with the same and diverged into two different mutations. Um, so people have actually been arguing over this for a very long time, whether molecular convergence even exists. So in Nature in 2013, there was a paper that appeared which argued for it, which means that they said because they observed a large number of convergent mutations which are shared by these mammals, especially in the hearing-related genes, they thought that yes, there is molecular convergence that underlies phenotypic convergence for a trait like echolocation. Um, but then a few years later, another paper came up in MBE which tried to disprove it because they said that when you compare it to control groups, they have exactly the same number of convergent mutations, and so there is no real evidence to say that molecular convergence underlies phenotypic convergence. Um, and so we sort of in our PNS paper we decided to sort of try and resolve this debate. And I'm not going to get into too much details, but we actually relied on two particular insights. One is that people were counting the convergent mutations, and that's not really correct. What you should be doing is asking uh, what's the particular function that they are enriching. Are they enriching like hearing function, or they are enriching like uh, uh, you know the size of the body, or height and so on. Like there could be a lot of these traits. Um, and In our case, what we see is that in case of bats, for example, they enrich hearing related functions. So if you measure it that way, you actually do observe that there is an enrichment. And also we actually tested for whether we think that those genes were actually lost, because sometimes what happens is nature just discards some of these genes and they are no longer useful in some of these species. And so what you see is that because they are under relaxed selection, so there's no selective pressure with them, so they acquire both convergent and divergent mutations at the same time. And that's one case that needs to be ignored. But bottom line, we were actually able to highlight for three convergent traits, specific genes, and the positions in which convergent mutations occurred. And we think, at least in our PNS paper, that we've definitely resolved, at least for some of these traits, this debate that was long-standing. And you can see over here that we had extensive media coverage even for this paper, including in Interesting Engineering and Science Daily. And lastly, I think like we also extended some of these techniques, like we used whole genome alignment, sort of like what I described in Segaline, to be able to discover gene losses. What I mean by that is, there are some traits which are a result of losses. For example, all mammals have hind limbs, right? Like most of the mammals have hind limbs. Like elephants have hind limbs, Humans have hind limbs, cats, dogs, whatever mammals you can think of typically have hind limbs, except that one lineage in mammals has lost the hind limbs. An example of that is dolphins and whales, which don't have hind limbs, as you can see here. And many people have wondered whether there's some gene loss, like a specific protein coating gene that must have been lost in these mammals that could have led to a loss of this trait as well. And that was one thing that was really hard for people to automate in the past. So they used to discover such gene losses manually, and that would take like several months to do it. And now, because you have a flood of genome sequencing coming in, can you actually automate that using whole genome alignments? And that's sort of what we did in our paper. I'm not going to get into details in the interest of time. But as you can see here, this is like a screen that we've developed, which involves a little bit of machine learning as well, like this outlier detection and so on. But the end result was that we have a completely automated pipeline that would run in a single day, for example, in a typical server. But with 58 mammals fed to it, it discovered hundreds of genes which were lost across different mammals. And we made a lot of interesting discoveries of genes which are, for example, associated with some of the human diseases as well, which are lost in some of these species. Um, and the good thing is people can actually now start doing experiments with these species and try and understand why don't they have this disease that humans have despite the loss of that gene. Uh, I'm actually going to skip over this, but there's a New York Times article which also said that there are some gene losses in bats, which could have led to because we think that coronaviruses they are opposed to a lot of coronaviruses, and maybe this coronavirus evolved first time in bats as well, and was then zoonotically transmitted to humans probably. And so this was an article that came long time back, but there are studies which have shown that gene losses also could also influence why bats live with so many viruses. And sort of our study, which was published earlier this year, sort of confirms this observation, in my opinion. So that's about my past projects. I hope that gives you a good idea, but I'm really excited about some of the projects that we're going to work on in future. So what's next? Number one, I'm actually moving on to UC San Diego. I'm going to be starting as a professor in electrical and computer engineering over there. As you can see, it's a really beautiful campus. By the way, this is the building, Jacobs building, which is probably where my future office is going to be located. I'm very excited to start there. But among the projects that I'm really interested in working on, one of them is actually genome graphs. So the idea over here is that we've been using a linear reference. Like, for example, I give you Bill Gates's example, that we could just compare our genomes with him and try and identify these mutations, which we have unique compared to him, for example. But this type of an approach we've been using for 20 years, by the way. And what we started to realize is that it has a lot of problems. For example, it's very biased. So, for example, Bill Gates is Caucasian, and I have Indian heritage, and we tend to have genomic differences um, which are profound, actually, compared to in ethnicities. We do often see that, and comparing cross ethnicities this way is not a very desirable approach. So what the consensus among experts that exists is that we should be using graphs as references. For example, we represent the sequences in the form of a graph, and so one path in this, for example, could be Vedant, one could be Yagya. Sorry to put you on the spot over there, guys, but this is just an example. And similarly, you could have one person, one female of an African heritage over here representing a different path, and so on. And you have a bunch of these mixed and diverse people represented in the reference. And that's going to be very important in our future analysis, but it also imposes a lot of computational challenges to do genomic analysis. For example, how do you construct this graph? How do you align now? All these algorithms, etc., need to be restructured. And this is one application where I feel that hardware acceleration is going to play a very critical role to enable these type of applications. So we're also already working on that. In fact, UC Santa Cruz Genomics Institute is one of the leading players in this area. And NIH gave a $30 million grant in which UC Santa Cruz is going to play a leading role. And I'm involved in that, and also involved with a lot of leading experts in this area. And I'm going to be continuing that effort once I move on to UCSD. But another revolution that has taken place recently is that of single cell genomics. And so now we can actually tear open a single cell and extract the DNA out of it and get the idea of the genomic sequence that exists. And with the help of that, we can actually now identify, for example, in case of cancer, what typically happens is that there's one or a few handful of driver, typically actually handful of driver mutations that leads to the cells dividing and dividing continuously, non-stop. But as they divide, they actually carry a lot of passenger mutations, and so it's very confusing to tell, what's a passenger mutation and what's a driver mutation. But with the help of this technology, you can actually do that, because you can actually construct the tumor phylogeny of each single cell, and then identify which mutations were actually driver. And this is also a very computationally challenging problem, where I think hardware acceleration would be really beneficial. And similarly, like I said before, we also want to study cell differentiation. Um, and so we are generating a ton of data to understand, which genes are expressed in what cells and what helps them make a muscle cell, and blood cell, and so on. There's a huge effort, billions of dollars are being spent on this type of research. And again, I feel hardware acceleration would be necessary to enable a lot of applications over here. And finally, there's phylogenetics. I already mentioned to you SARS-CoV-2 phylogenetics, but there's also a lot of comparative genomic applications. For example, vertebrate genomes are like huge. Virus is 30,000 bases, but our genome is, for example, 3 billion bases. So you can calculate that's 5 orders of magnitude bigger genome. So how do you do that type of analysis? The Usher is not going to suffice. And people have shown that even with few tens of species, it takes several hundred CPU years to actually carry out these type of studies, to carry out like building phylogenetic trees and so on, or to reconstruct ancestral genomes. How did the ancestor of human and chimp, how did that species genome look like? Or to do genotype phenotype association, like I showed for convergent evolution and gene losses, and so on. And so I'll be working on that as well. I'm very interested in that. And so with that, I'll actually end this talk, and I'll be happy to take questions. Thanks a lot for listening. But here's the detail of the lab. We currently have openings in the lab, and I hope some of you apply. If you have some past experience in this area, it's in these areas, it's ideal. Bioinformatics, computational genomics is not required. The above ones are in a way more critical. And I encourage you to apply for the PhD program at UCSD in the EC department by the December 16 deadline. And also, if you have questions about anything, actually, applying for PhD or applying to UCSD or my research, you can attend the next informal seminar that we have, or email me as well over here. So with that said, thanks a lot. I'm happy to take questions.
Right. Thank you so much, Dr. Turakhia, for this wonderful presentation. Now let us open the Q&A session for all the attendees. I would request you to please unmute yourself, then introduce yourself, and then ask a question. So please, anyone.
Hello, sir. My name is Anubhav Agarwal, and I have a question related to your Darwin project. So over there, are you using in memory computation in that SRAM part?
Yeah. So actually, people use and abuse this term in memory computing in a variety of ways. There's in-memory computing and near data computing and near memory computing and so on. Essentially, what I would call personally, and I think some may question that as well. I think that type of approach that we used in DSoft, which you rightly pointed out, that we moved the computation to SRAMs, that's a form of in-memory computing. Many people actually use it more restrictively, this term. They say that in-memory computing is more like, you are trying to move like some of these compute elements closer to like something like a DRAM type of memory. That's one form of in-memory computing. But I think the other form, and I think the more commonly used form of in-memory computing that has always existed in hardware acceleration, is the form that I described, which is to move some part of the computation and keep it in local caches, for example, and being able to manage that in a very specialized way.
Okay. Okay. Thank you, sir.
Yeah.
Someone just raised their hand. Can you please?
Hello.
Hello, good
Yes,
evening, sir.
Vipin, you can.
By the way, you guys don't need to call me sir. You can call
My
me by
name
my
is
first
Vipin
name.
Singh.
That's perfectly right. Yeah.
My name is
Yeah,
Vipin, and I had a doubt that how do you sample the DNA and process it?
yeah. So I have to point out that I'm a purely computational person. So personally, we've never really dealt with any of these squishy things that they have to really do. But there's a process that's involved in DNA sequencing. There is, you can start with uh, typically any cell these days. People do all sorts of things, but typically what people tend to use is saliva or blood. So the thing with saliva is um, it also carries a lot of uh, uh, other organisms like the pathogenic and so on. Blood is the more desirable approach. So you essentially draw in a few ml of blood. Uh, then what you have to do is like a library preparation. Essentially, what you're trying to do is you are trying to extract out high molecular weight DNA from the nucleus of that cell. Um, um, and there's a lot of chemistry involved over there. But essentially, once you do that, once you have those DNA fragments extracted out, the next step typically tends to be to amplify it, which is like the PCR step, which is what we are using in SARS-CoV-2 as well, the testing. They use RT-PCR, but in case of DNA, we tend to use PCR. We amplify it, which means we make tons of copies of it first. And then there are a number of DNA sequencing technologies out there. So there are two prominent ways to extract out like it's a very it's actually a very very fascinating technology that's there because we like in the EE world we tend to think of transistors and so on, like CMOS technology is so advanced. DNA sequencing is also very very advanced, because just think of it, the size of each base in the nucleotide is just like few nanometers they are apart, and we are able to precisely read that. And the way we extract out the signal, right, we are converting some signal into the alphabet of like a sequence of letters. The way we do it, we use two approaches. One is optical approaches and one is electrical approaches. So I'll tell you about the electrical first. One is the nanopore technology, which I mentioned, the portable device. Essentially, what they're using is they are taking the DNA, they are attaching it to a motor protein, and the DNA is sliding through a narrow pore. It's a nanopore, which means it's actually few nanometers in size. And as the DNA is passing through it, you're actually measuring the electrical current that's coming out, because each base A, C, G, and T have different electrical properties, so different resistance. And so you measure the current levels, and based on the current levels, you sort of estimate that this was an A, this was a C, and so on. The other approach is to do sequencing by synthesis, for example, where you take the DNA, you just take one strand out of it, and then you do synthesis of the other strand, which means you're making its complementary strand. And as you're building that, you actually attach when the nucleotide is being attached. Actually, they by the way have like custom nucleotides that are manufactured. But essentially, what happens is as soon as the base get attached, you release a light signal. And so, based on the color of the light signal, a green or a red, you can tell that it's an A or a C, and so on. I'm of course simplifying it grossly. Uh, but these are the two approaches. You actually convert some sort of a signal either through synthesis into an optical signal, or just pass the DNA through a pore, and that could be an electrical signal. And I have to say that this is another interesting challenge in hardware acceleration. Right now, base calling is also taking a lot of time. How to convert that electrical signal into sequence of bases? People actually use deep neural networks these days to do that. Um, and that has really changed the field completely, because the accuracy has improved from something like 70% to like more than 90%, even 95% now. Yeah, there's a lot of cool things happening at the intersection of CS and genomics. I hope that answers your question, by the way.
Hello, sir.
Thank you, sir.
Hello, sir. Good evening.
Yes.
My name is Shambhu, sir, from IIT Bombay. So the question that I have is like, you showed a graph where around 2025 there needs to be a species. I mean, there will be more species for which one needs to do a genome sequencing, which would take around three years, you said.
So not
So
sequencing, the assembly, the computational part would take 3 years for
yeah.
sequences which are sequenced in a single. Yeah, go ahead.
Yeah. So I mean, 5 to 6 years down the line, or maybe around 10 years. So if quantum computing comes into place, so where would this technology would be like? Would there be this technology ever there, or
Yes.
it would be completely, replaced by quantum computing? Like,
Yeah, that's
you see
a
the
good
future of
question.
this
Yeah.
technology? Yeah.
So there are two parts to this answer. One is you could call me a little bit of a pessimist about quantum computing, and that's actually for a variety of reasons. I'm not very closely tracked it, but I have interacted with experts in that area. And from what I understand is the technology it has always been for decades, like 10 years far. Everyone says that in 10 years it will come, it will come. It's actually fairly like we've not made enough progress, in my opinion, that we could say that in near term quantum computing, at least in the scales that we want to do it, the near intermediate scales, which people call, is going to be possible or likely in near term. And near term, I actually mean 5 to 10 years. I don't think that's going to come because there are so many technical challenges over there. Even though you may see like a lot of marketing being done there, like by Google, etc. That's if you get into the details, it's not. But there's another reason why it's probably going to be hard but interesting, and maybe that's another area of research which might be exciting for some of you guys. Is that a lot of these algorithms don't naturally extend, to quantum computing. That's how I understand it. I don't know the full details about it, but there are very few specific algorithms, and people have shown that for cryptography, etc., that you can accelerate through quantum computing something really, really fast. But I am not quite sure if people have actually demonstrated anything in genomic applications, like in alignment, for example. How do you do that with a quantum computer? Uh, so maybe that's a field of research that might be of interest to some people, but quantum algorithms is another area that needs to be worked on. And so far, I haven't seen much, at least in the space that I'm working on. Uh, I think, yeah, but I think in near term future, I think the sort of approaches which I mentioned, using domain specific hardware acceleration, I think that's uh, that's going to be very promising.
Thank you, sir. Thank you.
Yeah.
Hello, sir. This is Mihir.
I mean,
So when you talked about Usher, you showed us some specs, and you said you got like, so correct me if I'm wrong, you got like 100x performance improvement and sort of 10 times less memory. And you said
yeah.
that all this could be achieved by just improving software algorithms without going into hardware acceleration.
Yeah.
So could you explain that a bit? How do you get such
Yeah,
improvement
sure.
just?
Actually, that's another very good question. So, so actually, this is another way I would say is actually a domain-specific data structure optimization that we essentially use in this type of an approach. So just think about this: that the previous tools that existed, right? They existed not just for viruses; they existed for everything, and they didn't particularly exist for SARS-CoV-2. Even if they existed for viruses, let's say they were more specialized for that, they didn't exist for this particular virus. But what we are essentially doing is, so they were like very general purpose. So when you have something so general purpose, it comes with a lot of additional weight, that it has to generalize. And so it had a lot of overheads which were pretty unnecessary for doing something like a SARS-CoV-2 COVID-2 analysis. So let me tell you a couple of optimizations that really worked well for this virus. So number one, this virus is a slow evolving virus, which means that there are not many differences between any two viruses that you would sequence, even if they are like far apart in space or time. In fact, from the very first virus to any virus that you'll sequence now, the virus is first of all really small. It's just 30,000 bases. So that means 30,000 letters. That's 30 kilobytes. And if you represent it by bytes, you can actually represent it by every character by 2 byte actually. So it's really small, right? And besides being small, it has very few mutations. So typically from the very first till now, any virus that you sequence won't have more than 20 mutations. So that means you can really, really compress the information, the variation information that the sequences have into a very small representation. Now the other challenge is, so for example, if I have just one sequence, that's the reference, the first one virus, to now if we sequence, so I can just represent 20 times the number of characters, and those are the so it's like a few bytes of information that's there. But we have a lot of these sequences as well, right? And in fact, what we see is the other property that you can exploit is that a lot of these viruses that we are sequencing are essentially identical or share a lot of these mutations. So the very first mutation, let's say like a mutation at position 513, was shared by 5,000 out of 30,000 sequences. Then you can exploit that because you don't have to store again and again for those 5,000. You can just say that they are shared between these, and so you represent that information in a very compressed way again. And so with that type of optimizations, you can optimize the data object to be a few kilobytes, what was actually otherwise like several hundreds of megabytes previously. What other tools used to take those many megabytes for storing that information, we managed to store it in like a few hundred kilobytes to maybe a megabyte in size. So that was the number one optimization that we did. The other optimizations we did was related to like the search, which means that we actually encoded we pre-processed the data structures, which means we actually, within the existing phylogenetic tree itself, we annotated where we think the mutations have already occurred. And so when you're doing this search, you actually can just do a parallel search in which you are comparing to each point, and you already know what has occurred there, and you're just trying to do like a set difference. How different is this virus compared to that? And you are trying to ultimately find a min. So there was obviously a lot of algorithmic implementation challenges over there, but essentially once you do this pre-processing, the search is much easier, and so that gives you a huge improvement. And the other tools don't have a pre-processing object that they store. And so I didn't include the pre-processing time, but even the pre-processing time was not a lot. It was probably like 5-10 minutes of pre-processing time. But if you add that, then we don't have that much speed up, right? But we pre-processed it. So what I'm trying to say is to summarize basically the answer, we did everything in a very customized manner for this virus. We exploited certain properties that we saw, and by that we were able to make a very optimal representation suited for it, and that fits very well in the cache. And then you can do the search very efficiently on it. So those were the key steps.
Yeah,
By the way,
got it.
the speed was actually
Thank you.
more than 100x. I think it's closer to 3000x if I'm not wrong, because the next closest was 30 minutes and we were half a second.
Oh.
So it's a lot more, but of course I didn't include the pre-processing time. But we, in the server, for example, we've already pre-processed it, right? So you pre-process it once and you can sequentially add. So you added one sequence and then you can store another object with that just half a second. And then you can add one more to it. So you can do sequential sort of a thing. And that's less optimal than doing a global search, but it works pretty well in this virus. Does anyone have questions about PhD? Is anyone applying for PhD programs? Oh, hi.
Hi, sir. Yatish, before you go, so I'm Saravanan. I've been in the department since 2009. So I had a few questions about.
Yeah.
So I had a few questions about your numbers. So I'm not interested in genomics, but I'm more interested in hardware accelerators for cryptography and cryptographic applications. So the numbers you showed about this 40 nanometer TSMC, and you had shown this progression of the speed up from at every step in your first project. So was that fabricated or are those simulation numbers?
No, that was a design compiler analysis.
Okay. Okay. And if what would the cost be to actually fabricate such a chip?
Yeah, so I've been actually asking a few people about that. So we had actually interacted with one of the San Jose companies, which had showed a little bit of interest along with a VC to do something like this. We eventually ditched the idea because the technology is going fast. Sequencing technology is going fast, so it actually doesn't make sense to harden the circuit so soon. FPGA is a better solution, but they estimated roughly 6 to 10, something like that, $1 million for I think it was probably 28 nanometers.
Okay.
Either it was 20, yeah, I think it was 28 nanometers that they had said. It's a for 28 nanometers, I think it would be roughly 200 millimeter square chip.
Okay.
It was dominated by, but of course they included a lot of verification cost.
Okay.
On it.
So, so the FPGA, what would be the speed up? So you had about 30,000, if I remember, in the Darwin. So you said the Microsoft
15,000, yeah. Yeah, so in
15,000.
Microsoft research, so it depends how you compare. Now the power, if you do ISO, so this was ISO power, so it's 15,000 versus a single thread sort of a thing. Okay. We actually did at Microsoft, but we actually changed the comparison to a different tool, which was faster than that. It came up later. And we had like around like 100x speed up on the
Okay.
at Microsoft. It was actually Altera FPGA.
Okay.
It was ARIA 10. So it was
Okay.
a bit smaller than the Xilinx.
Okay. So you would
But
say
I
about
have to say that
100x.
yeah, but close to 100. Yeah,
Okay.
yeah, something like that.
Okay.
But I have to point out one thing: we did not manage to accelerate both pieces of the algorithm. So we did not accelerate DSoft. The reason is DSoft, as you had seen, right? It had a lot of memory requirement, so that
Okay.
doesn't fit onto the FPGA BRAM.
Okay.
So we ended up, what we did was we really optimized DSoft and software, but only managed to accelerate GACT onto the FPGA.
Okay. And what is the AWS F1? What is the average cost? I'm just curious. So you mentioned some
Yeah,
results using
so
AWS F1. Yeah.
So I think it's basically if you look at one FPGA that would come with 8 CPU cores,
Yeah.
that costs I think $1.6 per hour.
Okay.
And so if you compare it to a CPU instance, I think a 32 core CPU instance would cost you roughly the same.
Okay.
So you
Okay.
get 4
Thank
times
you.
lesser cores, but one FPGA. Yeah.
Okay. Thank you.
That's
And
not bad.
congratulations on all the great work you've done so far. It was nice listening to your talk and amazing presentation. I'm sure the students would have learned a lot from your presentation technique, the way you organize your slides. Anyway, thanks. Bye.
Thanks a lot. Yeah.
So, sir, are you inviting PhD applications only for students who are graduating next year, or for who are graduating later as well?
No, later as well. Like there will always be openings, I think, in the lab. So feel free to apply even later. This year, the lab is just starting, so I guess I have a few more openings in a way. So I'm definitely interested in talking this year's. But yeah, if you feel like you want to intern in the later years, join in the later years or do internships as well. Probably this year it won't be possible to give internships, but yeah, those type of things are also there's always going to be opportunities, I think.
Okay, sir.
Anyone else have any questions? So I have a question, Dr. Turakhia. You want to take some PhD students under you. So the prospective students, what they can expect, like what are the area of research in this? Some subtopics or something you can say about it.
Yeah. So, like I said, that I think this is my research is kind of so number one, I think I'm not really looking for people who are having any background in biology, bioinformatics, computational genomics, and so on. But I do want people to be motivated to learn, and hopefully, some of the applications which I showed um, uh, motivates you to actually work on it on something like that in future. Um, I would really like to have people who are really skilled in generally computer systems, and that could be especially people uh, who would be able to design hardware accelerators and who have very good understanding of FPGAs, GPUs, and so on. That would be an ideal fit, VLSI design, etc. Um, but have a lot of interest in learning biology because it's going to be a very interdisciplinary lab. As you saw in my last few slides, I actually did explain some of the future projects that I really am keen to work on. But in order to have any meaningful impact, I think you need to get deep into some of these fields. You need to understand, for example, like what is the genome really made up of? What are the different functional elements? What's our current understanding of what mutations cause what type of disease and so on, like how this analysis works. So it's a bit of an interdisciplinary research that you will have to do. And so that's kind of the expectation. In terms of the topics, like I said, that one of the things that I'm really interested in and I'm already working on is like this pan-genomic application, right? Like once we start incorporating all the diversity in the genome sequences that exist in humanity, like from all the races and ethnicities and as well as mixed ethnicities and races, you will have like a data structure that's huge but is very rich in the amount of information that it carries. And so I want to enable that sort of a thing. That's the current vision that a lot of scientists have, that we'll be transitioning to using something like that. and that's going to be one of the focus areas. The other focus areas, like I said, is that there's some sort of clinical applications as well. So, for example, like I showed you, with the help of like this new single cell sequencing data that's coming out, you can reconstruct tumor phylogenies, you can get ideas of drivers and passenger mutations and so on, uh, and be able to identify, what mutation is causing the cancer. And it's the first time that we are able to see a broad spectrum of mutations with the help of these long read sequencing technologies, etc. That you can make these inferences very accurately. So I want some people to be able to work on some topics like that. Of course, it will involve a lot of collaborative work. So it would probably involve getting a co-advisor as well, who would be able to collaborate and mentor on some of the complementary topics that would, or complementary areas that are required. And similarly, I mentioned some other like hardware acceleration work, whole genome alignments, comparative genomics, phylogenetics, including the pandemic. Actually, I'm writing a grant currently on pandemic, so some pandemic science. So in future pandemics, we are guaranteed to be doing the same thing that we did now, which is to sequence a lot globally. By the way, I didn't tell you that out of those 140,000 sequences, a lot of those sequences also are coming from India, which is very good to see. So it's truly a global scale now. And so if something like that doesn't happen in future, how do we track and how do we, uh, make use of all that information? That's going to be another research area. Um, so yeah,
Right.
that's just like touching upon some of them. But yeah, a lot of this would be also motivated by students' interest.
Okay. Okay. So can I ask how many students you will be willing to take? Is there a rough
Uh,
number?
so it's difficult to give a rough number. What I would say is I'll probably be starting not so large. So it's not going to be expanding to like 5 or 6. In the first year, it's going to be less than that. We'll start small, grow rapidly uh, in few years. Uh, but I think the stable size of the lab, I think if that's of interest, I think the steady state would be probably 6 students.
Okay. Okay. So we are about to come to an end. So if anyone has any question, we'll take one last question.
You guys are too formal. Someone needs to ask an informal question.
Yeah, so I'll just ask a general question. Uh, so from a student perspective, like I'm not sure like what I would be pursuing. If I'm interested in hardware acceleration, it has sort of different applications. Like you have hardware accelerators for ML specific tasks or edge computing. You have them in algorithmic trading, and so this is one of the new things that you mentioned in biology and genome sequencing. So what would be your suggestion, like how do I choose my field of specialization
Yeah, that's
in
actually a
this
very good
general?
question. Yeah, that's a great question. So the trick to choosing it, and I think I use the same strategy. Actually, I knew I wanted to do domain specific stuff, acceleration type of stuff, just when I joined Stanford, for example. And we had this rotation system, so you don't really come with an advisor or a topic. You sort of figure out everything once you join. Uh, but what motivated me and towards genomics, for example, and I think that's the sort of properties that you want to look for when you're choosing an application, is what I showed in my very first slide. And that type of slide, a very similar slide, was shown to me in one of the talks I attended. It was by the CEO of Illumina, actually, um, which does a lot of these sequencing. And what they showed is that, you have this Moore's law, and then you have like the data, which is growing much faster than Moore's law. I think that's the ideal candidate. So of course, genomics is one, like I showed you, that it's growing tremendously fast. Essentially, why is that important? Because it's creating a gap between how much you can compute and how much you can analyze, right? The computing
Right.
speeds are not keeping up with the rate of the data growth. And that's something that I show in all my applications, like I showed in comparative genomics as well, that there's a big gap that exists, and that somehow can be filled up with the help of acceleration, or can be at least mitigated with the help of hardware acceleration. So that's one property that you should definitely look at. And so machine learning is an obvious choice. AI is growing really fast. In video processing, for example, YouTube videos are also growing at a similar rate. There is, of course, astronomy and those kind of things are always like scientific computing applications, always going to be like that. But you never know with things like 5G, etc. What sort of other things people are going to do with their devices, with IoT. There's a lot of things people would do, and it would impose severe constraints. The other thing that you might want to also look at is we are talking about. You might want to consider is what has like these real-time constraints and so on. Like I said, pandemic has a real-time constraint, right? We want to get things done really fast. A hospital in an ICU analysis there, for example, neonatal ICUs, that will have that sort of a constraint. And if the volume of the data that's coming in and needs to be analyzed is very high there, then there I think you could, for example, require, because you want to do it efficiently, you might require hardware acceleration and so on. So you might want to look at the constraints as well. Like, do I really care about doing it fast? And how much does that doing it fast, what does it really enable? And so you really want to ask that question as well.
Right. Thank you, sir.
We have run out of time now, so let us all thank Dr. Turakhia for such an interesting talk. I hope some of us will be engaging our heads in it. They may try to help end the pandemic near future. Right. So thank you so much, Dr. Turakhia, once again.
Yeah.
Thank you.
Thanks a lot. Yeah.